Today we are going to discuss about the DNA replication. We've finished DNA transcription, where we found out that a protein is produced. Okay, transcription. But this is not transcription, this is DNA replication, and it occurs in the S phase in a cell cycle, the life cycle of a cell. S phase, where the DNA strand is still thread-like. It is not coiled to form the thick chromosome. Before this occurs, DNA is duplicated. So this duplication of DNA is called your DNA replication. So the process begins by separating the two strands. So this here is the DNA, and first of all, an enzyme called your topoisomerase, it latches on to the DNA. It latches on to the DNA, topoisomerase, and if there are any twists and turns or coils, the job of topoisomerase is to straighten that up. Okay, all the coils are straightened up, all the supercoilings are straightened up by the topoisomerase. After that, another enzyme called the helicase comes in and it begins to split up the two DNA strands. And it splits up this way and continues to move forward. You can see that this part, the DNA is still closed, okay? And it has this triple and double bonds, hydrogen bonds, that hold the nitrogenous bases together. And what the helicase does is it breaks up that hydrogen bond and opens up and forms this Y fork for replication. Okay? And as soon as this is opened up, you find that helicase opens up or splits DNA. But the DNA strands has this strong tendency to fall back and stick and form this double bond again. Okay? In order to prevent that, now that process is known as anneal or annihilation. So the word anneal is actually, it means, if you heat up something and let it cool, so that's the process of annihilation. Okay? But in bio, annihilation is, or annealing means the sticking of DNA strands. Okay, so that is what it means. So there's another protein called single-stranded DNA binding protein. Single-stranded DNA binding, that's a pretty long name. The work of the ssDNA is it sticks to the separated strands and prevents the falling back or the annealing of the DNA back into the closed DNA strand. Okay. So now, when this happens and your helicase is moving forward and this topoisomerase is moving forward, we find that RNA primer latches on to the portion that needs to be copied. So this is the RNA primer, the blue ones. Okay? Since we're talking about the DNA antiparallel property again, it runs from 5' end to 3' end, the top one, and the bottom one 3' end and 5' end. So in order to maintain this antiparallel property, 5' end comes on this end, and so the RNA primer latches on to this part. As it opens up, it latches on here and begins to, and this one is your DNA polymerase the work of the DNA polymerase is to get hold of your nucleotides and then stick them on in order to form this new DNA strand. Okay? So that is the work of DNA polymerase. And it begins its work right after where the primer is. So primer is the code from where DNA polymerase begins to build up this new DNA strand. Okay? So it runs from 5' end to 3' end this way. And on this side, on the bottom strand, we find it moving this way. On the upper strand, we find this moving the opposite direction, 5' end, 3' end. While these yellow stuff that you see, ssDNA binding proteins, they hold the strands from falling back. Okay, then we move on here. As the helices begins to move forward and it begins to open up more and more. So this is your primer, RNA primer. This is helices, which opens up the DNA. Here is one strand being copied, another one here. So this is 5', this is 3'. This one, this side is a 5', this is your 3' end. So let's move on. Let's say more of the DNA has opened up because of helices. Helices moves here. This part has been copied. Okay? And 5', 3', 5' here and 3' on the bottom one. Okay? Since this one, it has 3 and 5 here. So this is the reason why this strand is being copied continuously. In the case of the other one, you find it had copied this stretch. Now it opened up this much and it copies the remaining part. Then we move on here. We find that it has copied this much. Okay, this one again here. So it copied another one there. And as the helices moves forward, still further on, as the DNA is, separated, we find that it produces another piece with the primer here, and it begins to copy down this way. Okay. Whereas on the other strand, it has just one primer, and it continues to copy from the 5' to 3' end. So this is important to remember. On one strand, it produces fragments. On the other, it produces a long chain. This is the reason why we find during DNA transcription, and take you back to transcription, where we found that only one strand is copied. One strand is copied and the other one is not copied at all. This is mRNA. And the other strand is not copied at all. So this is your non-template and this one is your template strand. But in the case of replication, where the whole DNA needs to be copied, it has to copy both the strands. So in this case, as it moves down towards the right, one strand is being copied continuously, the other strand is not, because it runs from 5' to 3'. So pieces of it is copied. Now this piece was discovered by a person called Okazaki, a Japanese scientist. You may remember, it's easy to remember if you remember Shinji Okazaki. Shinji Okazaki is, he is related with winning the English Premier League with Leicester City. Okay? So Okazaki. These pieces are called Okazaki fragments. Okay, so here we are. The other one is copied at a stretch. So this one, which does not have any fragments, is known as the leading strand. And the other one, where you have opposite fragments, that is called, this strand is called the lagging strand. It's just that on the lagging strand, the copying or the duplication of DNA occurs in fragments, and it takes a little longer as compared to the leading strand. Okay? But in my diagram, you can see that the lagging strand has copied more, which is not the case. It's the opposite one, leading strand, which actually copies more of the DNA. That is just me. Okay. So, and then comes DNA polymerase 1, which converts the primers to DNA. If you notice here, there are portions here, blue portions, which are not the DNA, but it's the RNA primers, which actually give the DNA polymerase the cue, or it marks the place from where the DNA needed to be copied, okay? So, or duplicated. So these RNA primers are converted to DNA by DNA polymerase 1. And DNA ligase, now these are fragments, they're not joined, okay? And these fragments are joined together by DNA ligase. Joins the Okazaki fragments. So then you get this long chain of DNA on the upper side, as well as a long one which did not have any fragments at all. So two strands are duplicated that way. In replication, we find that only one strand is copied. Two strands are copied during replication, whereas in transcription, only one strand. And that strand is from the template strand. Whereas in the case of replication, two strands, one strand is called the lagging strand, which has fragments, Okazaki fragments formed, and the other strand is called the leading strand, where a continuous duplication occurs. So this is the difference between replication and transcription. This is DNA transcription.